It is my great honor and pleasure to give a brief introduction of Dr. James Rothman. He is the winner of Nobel Prize for Physiology or Medicine, and his research, which we will hear later, has transformed modern cell biology in a fundamental way. Dr. Rothman is an undergraduate at Yale University, majored in physics, which could be a hint that he would study how biological memory spread, cues, and move to support the daily life of every cell in nature. He got his PhD from biochemistry from Harvard, and did a postdoc with Harvey Knowledge at MIT. He has been a professor at Stanford University, Memorial Sloan Kettering Cancer Center, Columbia, and then Yale University. Before Dr. Rothman and Dr. Randy Sherman worked on the problem, we basically knew nothing about how vesicle inside a cell were transformed to the right place and fused with the right target. Using biochemistry, Dr. Rothman dissects every single step of vesicle trafficking and fusion. For the first time in the history of biology, we see molecular detail of how hormones are secreted, how cargoes in the cell are transported, and how neurons communicate with each other. For this achievement, Dr. Rothman won numerous awards, including being elected as National Academy of Science and the British Royal Society, Albert Oscar Award, and of course a Nobel Prize. Today we are more than happy to hear that Dr. Rothman is going to tell us how neurons talk to each other through synaptic transmission, the most fundamental step in brain function. Please join me to welcome Dr. Rothman.
And can you see me?
Yes.
Okay. Okay, very good. Ever since arriving here in Taipei, I've learned how to do this a lot. I'd like to thank the Dean for his very kind introductory remarks. It's my pleasure to be here with you today. I've had the good chance to hear my wife's lecture, which I don't always have a chance to do, just from time to time, so that added to the pleasure. And I'm beginning, it's my first visit to Taiwan, so it's been an important educational experience for me. I've been so impressed with the people I've met, so impressed by so much that I've seen, and I'm beginning only in a small way to now appreciate what a great country this small island really is. So thank you very much for making this trip possible and for welcoming us here so very well. Now, I'm now going to change the scale of the subject. So instead of talking about 2 whole brains or even 1 whole brain, what we're going to do is now change the scale and zero in on the smallest object of interest in the brain, which is the neuron, the basic parenchymal cell of the brain that is responsible for information processing. Next slide, please. And in particular, we're going to get even more focused than that. We are going to focus in on the synapse, which is the connection between 2 nerve cells that processes the information in the brain. In a real sense, the synapse is the heart of our intelligence. It's the heart of our central processing system. And in a moment, we're going to get even more focused in that we're going to look inside the synapse at the tiny little vesicles that store the neurotransmitter and the rules that govern the release of this transmitter, and therefore the rules that govern information processing in the brain. And shortly after that, we're going to even turn up the magnification higher, and we're going to look at the very molecules that are responsible for this process. And in the process of doing all of that, I believe you will agree at the end of the lecture that we are on the verge of discovering entirely new physiological mechanisms of neurotransmitter release that probably relate to different forms of learning and memory in ways that we do not yet understand. In other words, we will see how much it is that we actually don't know. But we'll do it armed and equipped with the very precise tools and technologies with which we can uncover important answers in the years to come. So let me begin by reminding you a little bit of cellular biology. A nerve cell has a cell body, and it has a long extension called an axon that functions as a wire. And you'll probably remember that in the cell body, typically, the membrane potential across the outer or plasma membrane can be shifted. And when that membrane is depolarized, meaning that the electrical potential is reduced from perhaps 100 millivolts to perhaps 70 or 80 millivolts, in a nerve cell, and it's a unique or nearly unique property of a nerve cell, an action potential fires, and that action potential or nervous impulse travels down until it reaches the end of the nerve cell, essentially the end of a wire. And so oftentimes the axon is analogized or thought of as a wire. It conducts the action potential from one end, the cell body, towards the nerve ending. It has a direction of movement. Now, there is a contact point, many actually, and in the brain, these synapses are much more abundant actually than the wires. So the synapses are the connections between nerve cells, and in the human brain altogether, there are about 10 to the 9th neurons, in other words, about 1 billion axons or wires, but there are 1,000 times as many connections as there are wires. Well, that makes sense, doesn't it? Because we want to be doing information processing up here, and it's the connections that process the information. So of course we're going to have a very high density of synapses up here, and the distances are very short, so the wires are very short. Now the meaning of that has an important meaning energetically. I think you all know that the brain consumes more metabolic energy than any other organ in the body relative to its mass, okay? And the reason for that is actually this huge number of synapses. Most of the synapses are not being used most of the time, but even though a small number are being used any one time, they're using up most of the energy. And most of the metabolic energy is spent on the process of synaptic transmission that I'll be describing. Moreover, the speed of information transfer, So this nerve cell is instructing that nerve cell to fire. This is a transmitter, this is a receiver. The synapse decides whether or not to transmit that information, essentially. And when because the number of synapses is so much greater than the number of wires, the speed with which we can process information is determined by the speed of synaptic transmission. So when we think about, and I'll come to this in a moment in more detail, when we think about what sets the speed of our central processing unit, our brain, it's actually the speed at the synapse, okay? So I will spend a lot of time today with you trying to ask the question, what sets the speed of the synapse at a very basic level? Because it's so very important for our ability to function as human beings. Could I have the next slide, please? So let's take a closer look inside of a synaptic connection. So now we've turned up the magnification on the microscope, and this is a nerve ending. This is the cell body would be somewhere up here. The axon ends here. So this is the transmitting nerve, and this is the membrane of the receiving nerve at the contact point, which is the synapse. You see the synapse as an asymmetric structure. You see these little vesicles in this cartoon. They're called synaptic vesicles. They're very small. Each one is about 40 nanometers or billionths of a meter in diameter. That's about 400 hydrogen atoms in diameter. They're very small vesicles. Each one contains about 3,000 molecules of neurotransmitter. Depending on the type of nerve or synapse, that will determine the type of neurotransmitter. So most synapses in the brain are excitatory, that cause the receiving cell to fire. They will typically have glutamic acid or glutamate as the neurotransmitter. Many, but a real minority, are inhibitory. They will instruct the receiving nerve not to fire. And a typical inhibitory transmitter is gamma-aminobutyric acid, or GABA. And there are many other familiar names. You have serotonin, which controls mood. You have dopamine, which provides reward. You have acetylcholine, which is important for attention in the central nervous system. And at a neuromuscular junction, which is what's shown here, in this case, the postsynaptic cell is a muscle, not a nerve. Then these vesicles contain also acetylcholine. It's an excitatory connection. So this is what these vesicles look like in the electron microscope. You see how uniform they are. Now, when the key point, as discovered by Bernard Katz and his colleagues in the 1950s, and which led to his Nobel Prize in physiology, was the discovery that neurotransmission relies on fusion of a single vesicle at a time. That is called quantal neurotransmission. And so the idea is that when a single synaptic vesicle fuses with this outer membrane on the presynaptic side, it releases its content entirely into the synapse, which is called the synaptic cleft. And it releases a fixed quantity of neurotransmitter, as I said, about 3,000 molecules. That means that a single vesicle will release always the same number of molecules of transmitter. And now we come to what those molecules do. On the other side of the synapse is the postsynaptic membrane. The postsynaptic membrane on the receiving cell contains receptors. For example, if this is a glutamate molecule, it will have a glutamate receptor. These receptors inevitably are ion channels. Most typically, they emit sodium ions, sometimes potassium, sometimes rather calcium ions. But what they serve to do is to depolarize the postsynaptic cell membrane to enable that cell to become depolarized, to enable that cell to fire. But it's usually a quorum decision. The number of channels that might open at a single synaptic point might be 5 or 10 channels. And that's enough to depolarize the cell, the postsynaptic cell, but not enough to make it fire. So typically, it's the collaborative effect of many inputs on that nerve, or many synapses, that represents how information is actually processed. So I think you can see that what is going on here, in broad terms, is that an electrical signal that carries information in the brain electrically is being converted to a chemical signal and then reconverted to an electrical signal on the other side of the synapse. And the reason that the body relies upon chemical transmission is that you can't conduct an electrical signal effectively through the water. And you can conduct molecules through the water. So now we have a simple elementary picture of how a synapse works. I've left out one important ingredient, and that's calcium ion. When the action potential arrives at the end of the nerve, there are concentrated here amidst the vesicles ion channels that are voltage gated. And these voltage gated channels open up when they are opened, but directly by the voltage from the action potential. They then emit calcium ions, calcium being very high in concentration normally outside the cell and very low in concentration in the cytoplasm. But the calcium ion concentration briskly rises in response to the action potential. And as you'll see, the calcium ion then triggers the fusion of these vesicles through an elaborate mechanism. Now, what is the mechanism that fuses these vesicles with the plasma membrane? That mechanism involves a process of membrane fusion that is actually quite general and involves a family of proteins called snare proteins, which we discovered many years ago. And what we discovered, and the dean alluded to this, is that snare complexes that form between two membranes are the general principle of vesicle membrane fusion. They, throughout our body, whether it's the release of insulin, proteins of vesicles that are involved in biogenesis of organelles and cell division, or of course synaptic vesicles, they all use this basic principle. And the basic principle is that two sets of proteins pair up between the membranes. So this could be the synaptic vesicle membrane, and this could be the plasma membrane. And you have one snare protein called the V snare, which is anchored in the vesicle, and it forms an alpha helix. And that alpha helix folds up very tightly with three other alpha helices that emanate from the target membrane. They're called T snares, for target snares. In this case, that's the plasma membrane. And this forms, as you can see, a four-helix bundle that literally folds up in this small space between the two lipid bilayers. And as it does, it pulls the two bilayers inexorably together. And as they are forced together, they fuse. And that opens a fusion pore, which in the case of a synaptic vesicle allows the neurotransmitter to exit. In the case of many other membrane fusions, simply allows the target membrane to grow by acquiring surface area and by acquiring new proteins. So this is the general principle of membrane fusion. It, of course, applies to synaptic vesicle release and therefore to quantal neurotransmission. Let's put this then, the release of synaptic vesicles, then into the broadest context in biology, and then we'll return to the special features of the synapse. Now, as I said, whether we are dealing with vesicles that are involved in secretory transport or endocytosis, vesicles that are involved in the release of insulin or digestive enzymes or other hormones or signaling molecules, or at nerve endings, all utilize snare proteins for membrane fusion. Different snare proteins, but closely related to each other and members of the same family. But there's a very important difference. Look at the speeds that are involved. A vesicle releasing insulin from the pancreas, where is my pancreas? Right up here. A vesicle releasing insulin from a beta cell in the pancreas, even in the fast phase of insulin secretion, even at its fastest, is 100 milliseconds, a tenth of a second. More often, it's slower. Enzymes that are being transported from their site of manufacture, for example, to lysosomes, are transported by vesicles that take several seconds, maybe even 10 seconds to fuse. So they take seconds. But in the synapse, they're so much faster. A typical synaptic vesicle will take 1 or 2 tenths of a millisecond to be released. In other words, 100 millionths of a second. And at the slowest, it will take 1 millisecond. This is what we call synchronous release. So the idea then, again, is that calcium enters and these vesicles release, and the new information I'm giving you is that it takes less than a thousandth of a second. If it were any slower, as I explained before, our processing of information would be slower. Now, actually, no matter where you look in the evolution of nervous systems, whether it's a person, okay, a worm, or a fly, the release of neurotransmitter vesicles, synaptic vesicles, will be this fast. It is a conserved property of evolution. It is actually perfect evolution, in all likelihood, to the limits of physics. It's actually limited, in all likelihood, by the rate of diffusion of proteins and the speed with which they can encounter each other in a lipid bilayer. So it can't be faster than this, and therefore we cannot think faster than this. It's a pity, though, because we might be better as a species than we are if we were only more intelligent. But we're not, so we have to live with what we have. But even here, there's a miracle, or what I think of as a miracle. Look at the difference in these speed. It's the same proteins providing the same amount of force that cause this fusion, or this fusion. Snare proteins in every case. And I'm telling you that over here, these proteins work 10,000 times faster than they do over here. Now, we all know, we're all familiar with car engines, okay? And we know, for example, the Germans, no offense to anyone here, continue to make the best car engines, okay? But I don't think that even the best German engine could run at 1,000 RPMs and also at 10 million RPMs. Can you imagine one engine and you shift the gear and it's 10,000 times faster? But this is the same engine and it's working 10,000 times faster. How can that possibly happen? Okay? So that is the question that I've been focusing on in my laboratory's research over the last 10 years. And as a result of a lot of patience and some good luck, okay, I can give you a bit of an answer today. I think a pretty reasonable answer, not at the level that I hope we will eventually succeed, but at a reasonable level. Now, what is it that we know and what is it that we don't know then about the synaptic vesicle? Because there must be something very special about it that enables it to do what I just told you at such a brisk speed. It's not the snare proteins. So here is a molecular view, a compositional view of a synaptic vesicle in an abstract way, and a small segment of the plasma membrane, of the presynaptic plasma membrane at the synapse, the nerve ending. In the synaptic vesicle are many proteins, but the most important ones and the most abundant are shown here. There is, of course, the V-snare, the helical forming protein that I told you about, that actually mediates the fusion, that happens to have this name, VAMP, in this vesicle. And then there's also, as you might expect, an ion sensor for calcium that can recognize calcium to trigger the release. And that calcium sensor protein also emanates from the vesicle. Its name is synaptotagmin. It was discovered actually by Thomas Suthoff, who discovered that it is the basic principle of calcium sensing throughout the body for membrane fusion, including the synapse. That's also why he shared the Nobel Prize with us the same year. Those are and then there's a third protein called synaptophysin. And this protein, until recently, had no known function. It's only our recent work that has elaborated its function. And it turns out that it's a very important function indeed. It is the key protein to allow these vesicles to fuse at the speed that they do. And you'll see that in a minute. In the plasma membrane, you have the snare proteins. There are two of them. One provides two helices called SNAP25, and the other is called syntaxin that provides the fourth helix to assemble the snare complex. And in addition, there are proteins that are called molecular chaperones, MUNC13, MUNC18. What those proteins do is they help the snare complex assemble. The snare complex, those helices, can assemble together spontaneously in the test tube, but in the body, they are sped up in that regard by these enzymes. So these are actually enzymes that assemble a snare complex. They are molecular chaperones that are different from the general molecular chaperones in our body in that they are specialized for these protein targets. And you'll hear a lot about MUNC13, which is the most important chaperone, in a minute. And then there are calcium sensors and clamps. And I've mentioned the calcium sensor, synaptotagmin, and there's another protein, I probably won't have time to talk much about it today, called complexin, which actually literally binds to the snare complex and prevents the snare complex from completing the zippering or folding process. And in doing that, it inhibits the process of fusion. And when calcium binds to complexin in a very general way, what happens is the complexin is released, and now the clamp is released. And so you can control or govern the process of fusion by applying sensors and clamps to control the process, okay? Now, it seems like we know a lot here. Actually, we do, okay? Actually, they don't give out Nobel Prizes for knowing nothing, okay? Right? But we don't know enough, okay? We know that these proteins are individually required. We know that there are human diseases associated with mutations in these proteins. We know that each of them is required genetically in model organisms for neurotransmitter release. But we still don't understand why it's so fast. Why then do we not understand this? Because we don't know how the proteins are arranged. We have no idea, essentially, or we didn't until the work that we've been doing, frankly, much of which is not yet published. We had very little idea in 3 dimensions how these proteins are organized. Are they actually organized precisely under the vesicle? Well, actually, we imagined they might be. Why? Because the easiest way to imagine that these snare proteins are able to do their job so much faster in the nerve than every place else is that they are better organized. They are perhaps highly arranged. Maybe they are actually arranged in a symmetrical structure that is highly cooperative so that they can all function together at the same time in a coordinated way. Instead of having each to go their own way, we all know that an army does better when everybody marches in step. And maybe these snare proteins are all somehow entrained in a structure to march in step. And maybe, actually, that structure was not discovered. We discovered this structure in 2019, not that many years ago. And we don't, as you can see, we don't have a very good picture of it yet, but we know it exists. So what this represents is cryo-electron microscope tomography of thousands of individual synaptic vesicles that are present in hippocampal neurons at nerve endings. With the work, as you can see, has been published. And what we've done is to average them in a completely unprejudiced way. We simply take what are called subtomograms and allow the computer to arrange them to look for the maximum correlations amongst them, making no assumptions whatsoever. And what we discover is that this is now a side view of the vesicle with the presynaptic plasma membrane, and this is a top view. You can see very clearly that there are 1, 2, 3, 4, 5, 6 blobs that are arranged in a circle and that they are perfectly symmetrical. That's even without assuming that they're symmetrical. Now, we can't see within the blobs at this resolution, and it's almost hopeless to do it in what was a living nerve. We are now trying to understand the structure, but as you'll see in a moment, we're doing it in a much more refined way where we have reproduced the entirety of the structure, we believe, in the test tube. And so we have a much more articulated system and precise system to study. But you can see the key features: six-fold symmetry and arranged in a circle. Okay, and the circle has a measurable diameter, so we know something about it. There's a regular structure. Surely the snare proteins are in there somewhere. Well, how many? Well, if this is correct, there has to be 6, or a multiple of 6. There could be 2 in each blob, or perhaps 3. But we know the size of the blobs, and it limits, the size is small enough that we can say at the most there are 2. So there are 6 to 12 snare complexes somehow hidden in here, along with the chaperone proteins, along with the calcium sensor. And if we knew exactly how they were arranged, we would probably understand a whole lot more about synaptic transmission. What we have achieved, so I've now told you what we have not achieved, which is a high resolution structure. But what we have achieved is we've learned how to reproduce the process of synchronous neurotransmitter release in an entirely synthetic synapse or synaptic system. And you'll see in a moment how we do that. But what we're doing is we're expressing and purifying each of the proteins that I identified for you and putting them into synthetic lipid bilayer membranes. And then we are observing these membranes to dock to each other, just like a synaptic vesicle docks in the nerve ending. And then when we add calcium in the test tube, these vesicles promptly fuse. And they fuse at the speed of 1 millisecond or faster. So we now know that we've achieved what we call the turbocharge of the synapse. Okay? We don't know how it works, but we've been able to reproduce it in the test tube. And that means we now have the tools to discover how it works. Because we've taken a problem of physiology, a very basic problem in physiology, and we have made it into a problem of chemistry. And a chemical problem can then be solved with chemical methods. And we've used these chemical or really biochemical methods over the last several years to begin to gain insights into how this process is organized. And those insights are summarized here. I won't have time today to go through all of the evidence. I'll give you some idea of the evidence. But first, let me tell you what we found so it will be easier for you to appreciate the evidence. We found that there are altogether, remember I told you from the structure, there should be 6 or 12 snare complexes. Actually, in this test tube system, there are exactly 12 under every vesicle that is docked and ready to release. And you'll see that we can actually count these molecules one at a time using single molecule optical counting methods under these reconstituted vesicles. And when we watch these molecules assemble, we actually see that there are 2 waves of assembly. First, 6 snares assemble as the vesicle sticks to the membrane. And then a second 6 assemble in a very precise process. 6 and then 6. And that corresponds probably to 2 waves of energy wells in an energy diagram. And what we know is that when the first 6 snares have assembled, and we call these central snare pins, when they have assembled, the vesicle moves closer to the membrane. The vesicle is first captured. You remember this molecular chaperone I mentioned, MUNC13? It actually has a series of very important roles. When it stands upright from the plasma membrane, it extends over a distance of 22 nanometers. And when it's 22 nanometers in extension, it first captures the vesicle. Then, once that has occurred, the the chaperone Monk 13, and by the way, there are 6 of them that capture the vesicle. So now you're beginning to see where 6-fold symmetry comes from. Each of these Monk 13s is able to make 1 snare pin. I told you it's an enzyme, it's a chaperone. One Monk 13 makes 1 snare complex. If you have 6 of them in a ring, how many snare complexes will you get? 6. You will actually use this Monk 13 molecule twice. First for the first set of 6, then for the second set of 6. I can't prove to you today that the same Monk 13 molecule is used for the first 6 as the second 6, but it's the simplest idea. But the fact that they are used twice is very clear. When the first 6 snare pins are made, the vesicle moves from 22 nanometers to about 14 nanometers away from the surface. How do we know that? Because we have an atomic level structure of this structure that forms that we call the lateral hexamer. And as you'll see, this Monk 13 molecule transits from an open configuration to a closed bent configuration in which it forms a precise 6-member ring. That is probably the origin of the ring that you saw at low resolution in the electron microscope, or at least in part the origin of it, and an important component of 6-fold symmetry. And then there is another rearrangement to a structure that we have not yet solved, but postulate. But we know there is a rearrangement which leads to this vesicle to being 3 nanometers separate from the plasma membrane. And at that stage, and only at that stage, does it have 12 snare pins. And at that stage, and only at that stage, can it be released by calcium. And when it is released, and you'll see this, it is released in the low millisecond range, most likely faster than a millisecond. Now, I'd like to come back to the 12 snare complexes. There are, we believe, 2 waves, and you'll see towards the end of the talk that we think that there are actually 2 rings of snare complexes, an inner ring and an outer ring. The inner ring is connected directly to the calcium sensor by binding it molecularly, and the outer ring is not connected to the calcium sensor. We call these peripheral snare pins as compared to central. Central is closer to where the fusion pore will open. Peripheral is an outer ring, okay? And the central and the peripheral snare pins will be linked to each other by the molecule complexin that provides the clamp. So I hope you're beginning to get a picture of what we're thinking. What we're thinking is that the central snare pins, which will zipper up first, they're closer to the contact point, they actually open the fusion pore in our model. The peripheral snare pins never have a chance to get there first. They're just too far out. What do? They provide extra force or extra energy to the 6 central snare pins. And I'll show you that when we eliminate the outer peripheral snare pins, these vesicles lose their turbocharge. They are the explanation, in part, okay, as to why the vesicles fuse so quickly. And I will also show you the molecule synaptophysin, the one that I mentioned that nobody knew what it did. It is responsible. Without synaptophysin, you will not get the second wave. It makes, without synaptophysin, you will only end up with 6 snare pins, not 12, and you will lose the turbocharge. So all of this adds up to a coherent biochemical, but not yet a structural picture of how this works. Finally, I would like to come to the calcium sensor. We've discovered that the calcium sensor, which was known actually to bind the snare complex, this comes from the work of Tom Sudhoff, who discovered with colleagues that the calcium sensor binds directly to the snare pin. And that was a very satisfying result, but it didn't actually explain how calcium binding by the calcium sensor leads to the release of the vesicle. Instead, the explanation seems to come from something that we've discovered more recently, which is that the calcium sensor self-assembles to form rings. Yes, once again, a ring. And you'll see these rings in the electron microscope. And these rings come apart when the calcium sensor binds calcium. And so what is going on now, what you're doing is looking down from the top of the vesicle, and I'm not showing you any other component, just the synaptotagmin molecule. There are probably 18 copies of synaptotagmin that form a ring. The snare complex is bound every 60 degrees on this ring. And when the subunits here bind calcium, when any one of them binds enough calcium, it destabilizes the ring, and the snare complexes are then released where they can complete their zippering. But you see, as hard as each of these snare complexes may be pulling inward as to zipper, it's actually, it is counterbalanced by a symmetrically arranged snare complex, so the net force is 0. That is why it is clamped, okay? This room is a symmetrical room. At its core, every building is an architectural monument to symmetry. It withstands the force of gravity because the internal distribution of these forces is precisely balanced by symmetry. I think you all know what will happen is if you remove a stabilizing element in this structure, the entire structure will collapse. Exactly the same happens with these synaptic vesicles when this ring is destabilized and the support system collapses. The result will be an explosive release. Now this slide simply illustrates Monk 13. I want to remind you that Monk 13 captures the vesicle in an upright configuration and transitions to a ring called a lateral hexamer. And I would like to now show you why we know this to be true. This is the structure determined by Kirill Grushin in my laboratory. We actually have two separate structures of this protein bound to membranes, actually high-resolution structures between two lipid bilayers. And what we observe is that in one configuration, which it will recycle, this is called the upright trimer. You see that there are actually 3 upright molecules that are assembled into a trimer. And when this recycles, you'll see that 6 of those upright trimers are arranged themselves into a hexagon. That is the first configuration that captures the vesicle. The vesicle will dock right there and remain here. And now this molecule will close through a simple intramolecular rotation, and that transits the vesicle closer to the membrane. We have crystal structures of both of these. We have made, I'm not able to show you most of the evidence, but we have made mutations that, for example, disrupt this interface right here in nematodes, and these mutations disrupt synaptic transmission. So we have this type of evidence both genetically and structurally to establish the critical role of these two intermediates. I'll take you back then to the structure that we saw under the synaptic vesicle in vivo with the 6 blobs. Now, these are individual rings of synaptotagmin, the pure protein, simply mixed with a lipid monolayer, and you can see they form very beautiful rings. From the diameter of the ring, we can judge how many copies of protein these rings have, and there are certain configurations in which we've been able to solve the structure by cryo-electron microscopy of these rings. And when we've done that, we actually can determine which residues are at the interface that stabilize the ring. We've been able to mutate those residues, and these mutant synaptotagmins actually are no longer able to assemble rings. So this means we can put the mutant synaptotagmin back into the nerves, which is what we've done over here. And now these are individual class averages. These averages get averaged to produce this. This is how that type of microscopy works. And I'm showing you the raw data, so to speak, so that you can see the evident 6-fold symmetry even with your eye before incorporating 6-fold symmetry into the analysis. Now, when you put the synaptotagmin mutant that will not form a ring into the nerve cell, you no longer see organized rings. Look, the rings are gone. What that tells us is the synaptotagmin ring is the organizing element that is responsible for this overall symmetry, even if we can't yet visualize the ring itself. Our resolution is not adequate, but we get rid of the ability of synaptotagmin to form a ring, and these rings whose diameter is in the same range go away. So that is one of the reasons why we believe there are synaptotagmin rings there, even though we can't directly visualize them. It's good evidence, but not proof. So now I want to take you to the functional reconstitution. I've told you rather insistently that we've been able to reproduce the process of synaptic vesicle release or synchronous neurotransmitter release, and I'm now going to show you some of that evidence. So what we do is we take these pure proteins, synaptophysin, the V-snare vamp, the synaptotagmin molecule. Each of these are produced in a separate test tube, and they are recombined with phospholipids mimicking the synaptic vesicle, and then we select reconstituted vesicles in the same size range as the native synaptic vesicle. And in the process of forming the vesicle, we allow it to enclose a dye molecule. The dye molecule is not a neurotransmitter, but it's our mimetic of the neurotransmitter. And we allow it to enclose a high concentration of a fluorescent dye. It's typically sulforhodamine B. And sulforhodamine B quenches itself. So at a high concentration, one dye molecule steals the fluorescent color from the other so that the overall color is reduced. And what does that mean? That means that there's enough color left so that we can see this vesicle, a single vesicle, in the light microscope as it lands on the surface of an artificial plasma membrane. We can see it land there, and then when we add calcium, we can easily see it fuse. Why? Because when it fuses, the dye is released, and the dye separates from itself, it dequenches, and the total fluorescence actually goes up until the dye diffuses away. And then for the plasma membrane, we produce what are called giant unilamellar vesicles. This is a different technique of membrane biology, which produces a vesicle that's 10 or 20 or 50 micrometers large. These are only 40 or 50 nanometers in diameter. It's so big, it's flat, essentially flat, and it's sort of like a plasma membrane, even though it's a big vesicle. And we include in this the natural proteins, recombinant proteins rather, SNAP25, syntaxin, and the chaperone MUNC18, along with critical lipids such as phosphoinositide and diacylglycerol, and that are required for the process. These GUVs are then allowed to sit down upon a microfabricated chip like you would use in a semiconductor, okay? And this semiconductor-like chip, which was devised, I'm sorry, could we go back to that? There we are, yes. Which was devised by Satish Ramakrishnan when he was in my laboratory, who had came as a biomedical and biochemical engineer. He microfabricated many hundreds of holes. So here you have a hole that's 5 micrometers in diameter. So the GUV is much bigger. It easily covers the hole. And in fact, I'm not showing you the next hole in the chip, and the next hole, and the next hole. So we allow this GUV to land. It then breaks open and it spreads. And it spreads upon and covers the hole. And then from above, we add the recombinant synthetic synaptic vesicles, which then settle down and they stick or dock to the plasma membrane. So you can begin to see here how this is just like the presynaptic membrane of the synapse, okay? And now the proteins that are engaging in vivo, they now do their jobs in vitro. And so we observe this by microscopy, and when we see vesicles that they land, and over 90% of those that dock are released when we add calcium. And as you'll see, they are released within 1 or 2 milliseconds. And as they are released, now shown here, the vesicle fuses and discharges through the fusion pore, the dye. Now, there's another feature of this system, which I won't have time to explain in detail, called evanescent wave optical microscopy, or total internal reflectance microscopy. And this is a standard tool that's used in single molecule microscopy. And we've arranged this chip, and it's been specially engineered so that we can also do single molecule microscopy. And with a special arrangement of the laser light, we're then able to see essentially throw a switch, and even though we illuminate the whole field with light, we only see fluorescence from 10 nanometers above the plasma membrane. So it's only when a molecule diffuses into the immediate range of the surface that we can see it, and therefore we see that signal above the background, which is essentially eliminated. That allows us to see individual molecules and count them. So now let's see what we see. Here we're looking at a single fluorescent vesicle. This is when it's self-quenched. This is the SRB dye inside one vesicle. The vesicle is landed on the membrane. We're looking at it from the top, and as a number, we're representing that vesicle's fluorescence versus time. Okay, so it's very steady because the vesicle's going nowhere. And it will sit there all day long. You can leave it for hours. It will do nothing. It's clamped. Now, you add calcium, and within 1 or 2 milliseconds, our time resolution in this experiment is 2 milliseconds of exposure of calcium. By the way, we have another dye here that glows when it binds calcium. And that dye is placed either in the vesicle or in the membrane nearby, so we can time exactly when the calcium arrives. Okay, we're not guessing. And you see that this vesicle fuses promptly. And you can see now we can look at hundreds of vesicles at the same time. So this is the real power of this method. And so in any one experiment, we get several hundred vesicles. And from that experiment, we can see that there's a very synchronous fusion of almost all the vesicles, not all, within the single time frame. And I'll now pick up the speed. We can count individual molecules of snare proteins under the vesicles. How? We simply take one of the snares, which is water soluble, tag it with a fluorescent dye, and we then look to see how much dye accumulates under one vesicle. This happens to be a single vesicle. We can see that the vesicle is there, and I'm not showing you this, from the SRB dye that's enclosed. But if we look in the other channel, we see at first the vesicle has no snares, even though it's docked. That's because it bound to upright Monk 13. Now, you see what happens is 1, 2, 3, 4, 5, 6 snares bind. And if we go to shorter time resolution, I'm not showing you that, you will see single steps. 1, 2, 3, 4, 5, 6. And then there's a delay. That delay, we think, but can't prove yet, is the time it takes the synaptotagmin ring to form. We have mutations in ring assembly that actually extend that delay. That's why we think that. And then another 6, okay? And we can take an average of many different vesicles, and we get about 6 and about 12, and I'll show you our most complete results in a second. And I want to come briefly back to synaptophysin, because it's very important. Synaptophysin, as I've mentioned, is the protein that makes sure you have 2 waves. How do we know that? I'll show you. Okay, so what we have here is this is what a synaptophysin molecule looks like in the cryo-electron microscope. It's actually a hexamer. It has 6 identical subunits, each has 4 transmembrane helices. If we mix pure synaptophysin and the protein VAMP, which is also present in the vesicle, the V-snare, just those 2 proteins together in a common bilayer, we can have them each with a different color, and then we can ask at very high resolution using turf microscopy, how often do these 2 colors come together in the plane of an artificial bilayer to form a molecular complex? And the answer is they do come together. And whenever they come together, there are always 6 copies of synaptophysin and approximately 12, 11 plus or 3, minus 3 copies of VAMP. That's because each of these subunits, and this was a known property from the work of Stowell and colleagues before our contribution, each of these protomers of synaptophysin binds 2 copies of VAMP, and that's because VAMP itself forms a dimer. One dimer binds to synaptophysin. So, what I'm telling you is, in the vesicle membrane of the synaptic vesicle, each synaptophysin forms a hexamer, and each hexamer sequesters 12 V-snares. Is that starting to sound like a familiar number? Is the number 6 showing up a lot? The number 12 showing up a lot? You're beginning to think there's something symmetrical going on, okay? So, interestingly enough, If you include synaptophysin in the reconstituted vesicle, which is the experiment I showed you a moment ago, and if you take all of the data that we have and average it together, you get 12 plus or minus 0.3 copies of the snare complex under every vesicle. But if you leave out synaptophysin, you now only get 6 plus or minus 0.3. So we know that synaptophysin is causing, it's not just that it's there, it causes 12 snare complexes to assemble. And by the way, why do we have 6 here? Well, you can guess by now. The chaperone itself, Monk 13, assembles into a hexamer. And how do we know that? Because we've made mutations at the interface that destabilize the hexamer, that eliminate synaptic transmission in nematodes, and we now get 0 snare pins. So the 6 that remained are templated by this hexamer. Okay? So there's no guessing here, actually. Okay? This is the power of this type of biochemistry. So now what happens to the process of membrane fusion when we have damaged it? Well, I think I've told you already that when you have synaptophysin, which is the normal physiologic situation that we're mimicking, you end up with 12 snare complexes and synchronous release of our neurotransmitter mimetic, right? Now, if you have left out synaptophysin, meaning that you have only 6 snare pins and it's a pathological situation, what happens? Let's look at this trace. This is just one vesicle. Does it look at all like this? Here's a happy vesicle in one experiment, synchronous release. Here's a vesicle that apparently is unhappy. Okay? It's from an experiment where it had no synaptophysin. It therefore contains 6 snare complexes. You see something completely different. Do you see the dye starts to dequench and does so progressively? And then it goes down. Okay? What's going on? What's going on, actually, is that the fusion pore, instead of opening all at once and releasing all the neurotransmitter, which is what Bernard Katz called synaptic quantal neurotransmission, is doing something different. 100% of these vesicles are opening their fusion pores and closing them over and over and over again. The first time they open it, they let a little bit of the dye out, and then they close. If they stayed open, all of it, even for 2 milliseconds or 1 millisecond, all of the dye would diffuse out given its rate of diffusion. But it doesn't. So it closes. And you see there are even indications of steps, which at higher time resolution than these experiments we see very clearly. So what's going on is this fusion pore flicker. It's been seen physiologically, but not commonly. Okay? But we would argue it may actually be a much more common occurrence physiologically. And this is what I meant. One thing that I meant when I said we might be discovering new modes of neurotransmitter release from this test tube system. I can't prove that to you today, but I think it's very likely. And if this happens physiologically, this is, by the way, the results of a population. So now you have a couple hundred vesicles averaged, and you can see there is no synchronous release. In fact, it's delayed by about 20 milliseconds. And then the average vesicle, in a gradual process, releases its neurotransmitter in microdoses over the better part of 1/10 of a second. Okay? So it's a completely different form of neurotransmitter release. Now, why is this? Okay, so if you might be thinking that, what I'd like you to think is that the turbocharge requires 12 snare complexes rather than 6. That's one interpretation. Because you have 12 here and you have 6 here. Here you have fast release, there you have slow release. So a simple conclusion is the turbocharge comes from the 12 snare complexes as compared to 6. But there's actually 2 variables in this experiment. The other variable, in addition to the number of snare complexes, is that there's, chemically speaking, no synaptophysin here. Maybe synaptophysin is the key ingredient. Well, what would be the ideal way to distinguish between these two? It would be if you could add synaptophysin, but the synaptophysin would only assemble 6 snare complexes, not the usual 12. What we found is a mutant synaptophysin from a patient. So this is a gain of function mutation that is was found in a little girl in Iraq, actually, by my collaborators at University College London. And this patient has a series of typical synaptopathy symptoms, including intellectual disability, slow development. They're listed here. And that thus came to the attention of the neurologists, and thus ultimately to the attention at UCL. And they sequenced her genome and found that there was a newly acquired mutation, not present in the parents, therefore highly likely to be causing the disease. And it was in synaptophysin. And so it turns out to be an amino acid change in one of the transmembrane domains. Just one helix, one amino acid, a single amino acid change. And when we took this gene and we expressed it in tissue culture cells, purified the synaptophysin, what we found, and unfortunately you can't see this here, was a completely different pattern. Okay? This is what we saw with the normal human synaptophysin, and this is what we saw from the patient. Exactly this. So the conclusion then is that, and I left out one other very important things. We redid the experiment. Remember the experiment I showed you where we counted the number of snare pins and counted the number of, remember the experiment where we put synaptophysin together with the V-snare vamp in the lipid bilayer and it bound 12? This altered synaptophysin from the patient only binds 6. So we have synaptophysin there except for one amino acid out of 300, okay? And it's about the best you could hope for. And you now have 6 snare complexes. We don't know why yet, okay? But it's a fact. And when we have 6 snare complexes, we have reduced, altered synaptic transmission. The turbocharge is there. And that is why, and I'm going to now finish up rapidly because I know I'm going over time. That is why we believe it is these outer snare pins. This is a structural model based on the synaptotagmin ring and some earlier X-ray crystal structures that I'm all sort of putting together, taking a bunch of structures that are known and putting them together like a Lego kit, okay? But without going into the details, the central snare pins and the peripheral snare pins connected by complexin, what we think we're doing is eliminating these, eliminating the outer ones. When we consider the physics of this, as has been done by my colleague Frédéric Poncet at École Normale Supérieure, who is a soft matter physicist with whom we collaborate, he points out to us that the peripheral snare pins are not in a position in our structure to exert force on the contacting membranes directly. They are only in a position to pull the outer membrane of the vesicle downward, okay, downward. And when they do that, they are only in a position to actually increase the pressure inside the vesicle. So if you reflect on the physics of the matter for the moment, if you pull on the sheath of the synaptic vesicle to flatten it, you will be applying force to the water inside, which will apply, according to Sir Isaac Newton, an equal and opposite force pushing outward, which we call pressure. And he's calculated approximately what that pressure is. It's about 3 times the atmosphere. So if Fred is correct, that pressure, that additional pressure from the peripheral snare pins explains, and he can predict quantitatively actually, the enhancement in fusion pore opening rate from 6 to 12 snare pins. That gives us the turbocharge. The rate of fusion pore expansion, making it a one-way expansion. In other words, he's predicting that with 6 snares, you'll get fusion pore flicker. But when you have the extra turbocharge or push from the peripheral snares, it will push these fusion pores open in a one-way journey. And therefore, you never get the fusion pore flicker. And most interestingly and importantly, when you open up a hole like a fusion pore between a high pressure content and low pressure, meaning atmosphere pressure outside, what will happen? I think I'm describing to you a garden hose. You have high pressure inside the hose, and now the garden hose, what does it do? It ejects the water. And so you will be ejecting water. And he's calculated that it will take only about 200 nanoseconds for this neurotransmitter containing water. If this is all true, it's just prediction. But if it's true, it suggests that there's an entirely new mechanism of synaptic transmission in which the neurotransmitter is actually not diffusing out, but is actually ejected, it is sprayed against the postsynaptic membrane with almost no dilution. And that has tremendous physiological implications if it's true. We don't know that it's true, and it's very hard to test, but we're trying to develop ways to test it. I just want to recapitulate what I've told you because it's been a long talk. The idea is what I've been talking about here is a supply chain for ready-release synaptic vesicles. So the physiologists refer to the one or two synaptic vesicles that are released by a single action potential within a millisecond time frame. They are termed ready-release vesicles, and they are said to constitute the ready-release pool. This pool of vesicles has to come from somewhere. It comes from a supply chain. I have outlined you our current best insight into what this supply chain consists of, in which when this vesicle fuses, it needs to be replaced by vesicles in this stage, which we call mobilizable. In fact, a mobilizable pool has been demonstrated by Erwin Neyer and colleagues. Its molecular substrates are not known, but it takes 10 or 15 milliseconds to mobilize after calcium. And it is known to be present at about 10 or 15 nanometers distance from the plasma membrane. So that fits very well. And there's another population which we believe exists that is the precursor to this population. Ultimately, it is the calcium ion that mobilizes all of these vesicles to allow each to replace its successor along the supply chain. And these waves involve, as you've seen, successive waves of assembly of snare complexes, 6 at a time, to produce a ready-release vesicle that has 12. And it appears that it is the number 12 that is the key answer to what makes this vesicle as fast as it is. I hope you'll begin to think of the process of release of neurotransmitter as an explosive release, not unlike a bottle of champagne. There is a lot of energy that's stored in the snare proteins. It's stored in a container similar to the champagne bottle, but the container is actually made of this chaperone Monk 13. Monk 13 gets closer and closer, so as the amount of energy that's stored goes higher and higher. Each time 6 snare pins are made, the amount of energy doubles. It doubles in a volume that's smaller and smaller. And if you have more and more energy, next slide please, in a smaller and smaller container, you are of course adding pressure. You're creating pressure just like inside here, okay? And as that pressure builds up, and if you release it fast enough, and it is the release of this stored energy by calcium when the synaptotagmin ring dissolves, that is equivalent perhaps to the release of this cork. And so I think we now think of the process of neurotransmitter release as being qualitatively different from every other membrane fusion in that it has an explosive character because it invests such a high amount of energy in such a small volume and uses such a brisk trigger. Thank you very much. And I'd like to just call your attention to the people that have done this work. This is not an overnight, I know you're going to come up with something, just give me one sec. It's very important to acknowledge the people that have done the work. And this is not the work of a few years. I am now summarizing 10 years of work. And we're not even yet not at the answer. We still have the cryo-electron microscopy of these reconstituted membranes to do, which is what we're hard at work on right now. The development of the cell-free system involved actually 2 generations of people in my laboratory. Shyam Krishna Kumar and Satish Ramakrishnan are both independent assistant professors now at Yale. And they developed the initial stages of this cell-free system, and Satish developed the chip. Their successors in the lab, Atruli Chatterjee and especially Venkat Kalyana Sundaram, developed the system into its current state of maturity. That was, by the way, 5 solid years of work on top of their 3 years of work. And in the last 3 years, we've begun to learn the many wonderful things that I've suggested to you, which are just really the tip of the iceberg, especially through the work of Manindra and to a certain extent, but an important extent, Atruli and where's Feng Li? And Feng Li, who have done the single molecule counting studies that I described. And we continue to develop an understanding through cryo-electron microscopy. Kirill Grushin, in particular, discovered the oligomeric states of Monk-13. And Sudhanshu and Abhijit are making great progress now in getting high-resolution structures of the interfaces of these various intermediates. And it was actually Abhijit in his earlier work who discovered the 6-fold structure that at low resolution in the nerve endings. And then we have a number of important collaborators at Yale and elsewhere, but I'll especially mention Jeremy Dittman, who is a neurobiologist who studies C. elegans at Cornell, with whom we've collaborated on all of our physiologic studies involving genetics, and Kirill Valinsky, a neurophysiologist at UCL, with whom we've done physiologic correlation studies for the synaptotagmin ring, and now continue to do for measurements of subquantal transmission, evidence for which is now emerging rapidly in his hands. So thank you, and I should mention again Fred Poncet. Where is Fred? Fred Poncet, who I highlight up here because we have had a 15-year collaboration, and frankly, he deserves as much credit for most of what I've described as my own laboratory. It's really been a very deep and enduring collaboration that has involved both physics and biology. So thank you very much.
Opening to a new concept. Let's open the question to the audience. Professor John.
Yes, thank you very much for the very stimulating, beautiful talk. So if I understand correctly, I think there are hexagons formed by the V snare that's on the vascular surface. And I would tend to think that there are also hexagons on the plasma membrane formed by the T snares. And when the two encounter each other, this initiates a membrane fusion. So, but my question is that for the vascular surface, the membrane has a very sharp curvature, but for the plasma membrane, the surface is kind of flat. So I wonder if there are fundamental mechanistic differences between the assembly of these two types of hexagons.
Okay. So that's you're bringing out, I think, that's a very good question and a very good point. The structures that we have demonstrated actually form very well and uniquely well when the vesicle is of the diameter of a synaptic vesicle and when the plasma membrane is essentially flat. And we're starting to find actually that when we, just for example, when we reconstitute vesicles and have a wide range of diameters, it's probably the vesicles that are most mimetic of the synaptic vesicle that form ordered structures. And when I'm now going a little beyond our data, but it's from the way it looks, when you have vesicles that are larger, they will form they will form actually 12 snare complexes, but they will not be probably arranged in they will not be in a completely ordered structure, which requires they come together in rings with a radius that's compatible with the radius of the vesicle. When the vesicle gets too big, for example, the Monk-13s, the chaperone that's on the outside, gets pushed out too far radially. So, I mean, I can't go into all the details of that, given this, I'm happy to talk to you later if you want. But the diameters are really key. And I think they actually fit together like a lock and a key. You know, there's almost nothing about this system that turns out to be by accident. I think this is such a highly evolved system that is evolved, as I said, in an almost perfect way, that we're just seeing the tip of the iceberg of the type of molecular choreography that exists here. And as this mechanism unfolds, I think we're going to see a symmetrical dance of protein molecules involving multiple states. And let's Monk 13, for example, has in its upright state, we actually have yet another 2 states that we don't understand the full physiologic meaning of yet, but they're transition states that represent intermediate states of ratcheting of this molecule. So every single thing in this dance is prescribed choreography. And I don't doubt that the diameter of the vesicle is a very, very key aspect.
The question is why these 2 things, size, are similar and how they coexist and function. The synaptic cleft and the vesicles.
Synaptic cleft and vesicle.
Yes, because the answer on our paper is both 15 nanometers.
Okay, the synaptic vesicle is about 50 nanometers, that's right. And the synaptic cleft. Oh, I see. I mean, the width, the gap of the synaptic cleft, why are they similar in dimension?
Yeah, because
I simply don't know the answer to that. You know, it's funny, I never actually thought to ask the question. And maybe it's not an accident, actually. I actually showed a picture of a neuromuscular junction because it's a sort of famous picture in neuroscience that I clipped out of a textbook. But the neuromuscular junction is much more extensive because than a central nervous system junction, which is called the latter is called a bouton, and the bouton is typically about a half a micrometer to a micrometer in diameter, whereas you have a much more extensive string of boutons that almost are continuous to allow a much larger amount of neurotransmitter to be painted onto the much more massive multi-nuclear muscle fiber. A muscle fiber is much larger, and they're single cells. So you need to have much more depolarization to have an overall effect. Did I get that right? Okay. Okay. Good. So I expect extra credit from you. Okay. Good.
Hello. I have some idea for the last part of that narrow torrent from the from your talk, and maybe you can try to introduce some ion current in that system, because all of the systems are man-made in your research. So maybe you can introduce some kinds of ion current, and then you can measure it in the ampere or like the voltage. Then you can and you also know how wide of that tunnel is. So then you can calculate that.
Okay, that's fantastic. Okay, that's exactly what we're doing.
Oh, really?
Yeah. Yeah, thank you. You should come and work on it. What we're doing, actually, we in this case is Dr. Ramakrishnan, in his lab, which adjoins mine, is introducing. He's taking the chip that I just described and building into every well electrodes, and then we're putting an ion channel, okay, really a hole in the membrane. Can we go back a slide? Oh, I don't want to go the whole way through it. No, never mind. But we're putting a channel in the vesicle, so that when the vesicle enters the plasma membrane through fusion, or even forms a fusion pore, you now have an electrical conductance. And just as you suggested, from the magnitude of that electrical conductance, you can measure the diameter of the fusion pore, and you can measure that directly. And the time resolution in doing that now moves to about 10 microseconds. So we've pushed optical resolution beyond really what any lab has done with down to 1 or 2 milliseconds. But that technique will allow us to transition to the point that we can measure the precise properties of that fusion pore and probably observe the openings and the closings and try to understand how that is physiologically regulated. And I personally have no doubt of the physiological importance of this. Thank you. It's a good suggestion. Good work. Okay, this guy should get extra credit. Okay.
So, but before we close the important symposium today, we are going to welcome our President of National Taiwan University, Dr. Chong Wen-Zhang, to the audience to give few words and also give the trophy to Professor Rothman.
First, I would like to welcome Nobel laureate Dr. Rothman to visit the NTU and give a speech, and also that Professor Hirsch. I think I missed her talk. I'm very sorry because I had a lot of schedule. I have to meet today. But anyway, I believe all the audience enjoyed the speech from Dr. Rothman. I can see that there are some questions when I come to this lecture room. But this is only the beginning. I hope you can come back again to have a communication with our College of Medicine and also the university. I believe that next time we can invite you to, you should be in our main campus. I believe there are some College of Life Science and other colleges. They will be interested in your speech. And besides that, the college, we have a department, a nursing department. The year, I think that the College of Nursing, they just visited me last month. We talk about the possibility of the collaboration. And it looks like very promising because we have a Yale alumni there. But anyway, really appreciate for your speech and hope that we can have more future collaboration or communication. And also welcome to Taiwan and NTU. Thank you.